phenology and the challenges that you've faced. I'm sure it's going to help us a lot in continuing our work. So we have the third speaker, Dr. Suresh Hedges. He's a consultant scientist at the Center for Ecological Sciences, IISc, and he has his PhD from Forestry Research Institute, Dehradun. He has also worked with Dr. Mohan Varghese of Institute of Forest Genetics and Tree Breeding, Coimbatore, on phenology of tropical Montana forests. Dr. Suresh is a trained forest botanist, and he's been monitoring the phenology of trees in the 50 hectares and 1 hectare permanent forest dynamic plots established in and around the forests of Madumalai for the past 30 years. He's also worked in dendroecology for dry and coniferous forests of India, and his research interests include phenology, biodiversity, climate change, dendrochronology, and several others. And he has published over 100 research articles in both the national and international journals. Dr. Suresh is part of the Global Network for Forest Permanent Plot organized by the Center for Tropical Forest Sciences, CFTS, and he's also the trustee for the Asian Nature Conservation Foundation. He's been actively involved in other research activities on plant ecology studies that's conducted at CES IISc. Welcome, sir.
ಇದನ್ನ ಕೊಡ್ತೀನಿ ಸರ್ ಫ್ಲೈಟ್ ಸೈಡ್ ನೆಕ್ಸ್ಟ್ ಇದನ್ನ ಮಾಡಿ ಕಾನ್ಸೆಪ್ಟ್ ಮಾಡಿ ಇದು ಫ್ರಂಟ್ ಫಾರ್ವರ್ಡ್ ಇದು ಬ್ಯಾಕ್ವರ್ಡ್ ಇದು ಬಂದು ಪಾಯಿಂಟ್
Thank you. And it was a very beautiful introduction by Dr. C.P. Sen Rajay, which made my work very, very easy. What I'm going to do now is, this is purely a ground observations, and what we can capture from the Phenocam or satellite, and what we cannot capture them from these Phenocams and satellite, and what is the necessity for ground observation. I'm going to stress on that a little bit. And this is the data from dry forest in Madumalai. Yeah. Yeah. So a little bit of introduction, which is may not be very much required here. Dry forests are characterized by seasonality in climate, and then the moisture stress and interannual variability in rainfall are very, very important in the dry forest ecology and all that. And the moisture stress is variable with 3 to 6 months of dry season on the global level, and seasonality is mainly driven by the rainfall. The droughts in these areas are very common, and each species has an individual adaptation for combating the drought. And then to capture all that, we require to study them. And phenology is just the science of appearance. If you look at an individual, how does he's looking at, or how is he appearing? That is one. And it is the phenology, especially of the dry forest, is very, very important or very interesting, because many a times it is linked to the climate. Then the responses that you observe in the plants is the outcome of environmental conditions. Now, how the plant is subjected to the different kind of environment conditions, then plant responds. And then the global climate change that we are observing, increasing the temperatures or changes in the rainfall regimes, is affecting functioning of various biomes and various ecosystems, so to say. And then it is the plant phenological response is one of the most sensitive to the parameters assumed to alter because of the climate change. And then long-term quantitative plant technology will be very much accessible. That would provide something of a baseline information to model various impacts of climate on the dry forests. And it's a potential tool. Yeah, I think I will, is this not the plant responses? It is also associated with its processes like carbon sequestration, reproductive output, and it also affects the coevolutionary factors like pollinators and dispersants. This study comes from a dry forest in Mudumalai Tiger Reserve. This is part of the large Nilgiri Biosphere Reserve and the first of Biosphere Reserve in India. And there is a distinct rainfall gradient in the steady area, the wet being the eastern portion of the center being dry and west being wet. And then we also monitor the climate in the state. And our Mudumalai has been the long-term research guide for our center. So this is the study area where we also have a large 50-hectare permanent plot, which we are part of CGF network, and this is the overview of the dry forest that yes. This is the dry forest in I'm going to just show you very briefly about the different kinds of forests that we are talking about. This is the dry forest in Mudumalai. This is the dry residual forest, and this is moist kind of forest. This is how the 50-hectare plot laid on the Google imagery, and this is the plot.
Yes, long press.
Long.
Long press. Yes.
I can operate in June. Here it's
First slide.
No, I mean the various photographs. Okay, next up.
You know. Mouse, mouse. Open up the sorry.
Any problem?
Yeah, I just checked that. Can you give me that pen drive? Pen drive.
Okay.
Which one? This
Is it EMPRI?
EMPRI. Okay. Okay. Is that right?
Yeah.
Okay. This is the then was it?
Even.
So it was it. Yeah.
Yeah.
Sorry.
No, no. Yeah. I'm sorry for the interruption. Yeah, this is the long-term study sites from CTFS across globes. We have all the forest plots being enumerated in a uniform method so that the results and the data can be comparable. So what are the methods that I'm following in Mudumalai to monitor this phenology? We have about 335 reproductively mature trees belonging to 52 species with very clearly visible canopy. We are monitoring them. Monitor for both vegetative and reproductive phenology. The extent of each phenophase on the canopy of the tree was qualitatively measured. Then we used a measure called intensity that Dr. Sharma was describing as a weighted measure of both proportions of individuals, extent of canopy, and number of species participating. That becomes a more quantitative estimate of particular phenophase. And then we collect climate data from our weather station installed on site called Kalgudi. Then, this is, we started these observations from 2000 and the phenology monitoring is going on since then. Now we have increased one number of trees. So interestingly, between 2000 to 2004, there was a very big drought in which the normal or the mean annual rainfall was less than 40%. And then between 2004 to 2008, that was an abundant or a normal period of rainfall. So that it gave us a natural experimental setup for us to compare the phenological patterns between two different periods. How does it impact on individual species? This is the plot of the rainfall in Mudumalai since 1990. I plotted till 2017 or so. And this is how the mean monthly rainfall looks like. And this is the mean monthly rainfall during the drought and the normal period. Between 2000 to 2004 was a drought, and then 2004 to 2008 was an abundant period. And then this both the amount of mean annual amount of rainfall plus the number of wet days was statistically significant between the both periods. So whatever the results that or whatever the inferences that we draw has a validity. Now, if you look at how does it at the community level, how does the leaf initiation looks like? This is over 8 years I've plotted this. So there are distinct peaks and then there are distinct layers in both initiation and leaf expansion. Okay. And this is what happens with fruit initiation and flower initiation in the Mudumalai. Yeah. And how does this happen with if you plot now both drought and the normal period? The drought period is represented as line graph, and then the normal period is the bar graph. Though the pattern looks almost kind of similar, you just look at the intensity of that. During normal period, the more number of there is bigger or the greater intensity of initiation compared to the drought, and also in the expansion part of it. This is with the young fruits. This is the flowers, both initiating flowers and mature flowers. And this is with the young fruits and mature fruits. Okay. Now, how does an individual species respond? I have taken 4 canopy species: Tectona grandis, Lagestromia, Terminalia, and Anagasis. Why I took 4 of these species? These 4 species together accounts for about 60% of the total abundance. So any inclusions or any inferences that I draw will have a big impact at the community level and at the canopy level. So what I've done is I have plotted the initiations of leaf, okay, between 2 periods, the drought and the normal period for each of these. So the red line represents the intensity of leaf initiation during the drought period, and the blue line represents initiation in the normal period. So if you observe, there is a very strong differences in the intensities. The strength of the intensity in the normal period is much more higher than during the drought period. Pardon? That was the analysis. Yeah.
Can you say based on the graph that analysis is more resilient to drought?
Probably you may be, so you may not be on the right path, because though the pattern looks kind of similar, Anagasis has twice 2 peaks bimodal wave, but if you look at the intensity of that, during the normal period, the intensity is much more higher than the drought period. This is how the individual species respond.
If I have to draw inference in terms of resilience, drought, showing welfare.
In terms of that, probably you are correct. While Anagasis, has much more resilience compared to both or compared to all the other species. Yeah, because Anagasis is typically a very dry forest species. Now, if you look at all of them, this is more interesting. Okay. Suppose now you put all the initiations during the drought. What I want you to observe is in the first panel, during the drought years, you look at the extent of leaf initiation, the number of or the amount, the months that each species show the initiation. While in the normal years, the process would have been completed, and then they're much more synchronous. This is very important from the point of view of energy budget. Now, each phenophase or each stage in that is highly energy demanding. For example, if you have to initiate a leaf, the tree has to spend a lot of energy. And then if you keep on spending your energy, then where do you store it? In the sense, you don't put that energy in the production of wood, which means that your sequestration of carbon goes down during the drought years compared to the normal years, because you would be spending your time and effort and energy in putting up your leaves. Now, if you come to the flowering season, it's almost kind of a similar pattern where the intensities are low, and then Anagasis has sort of shown again some kind of a resilience, because it's basically and ecologically adapted to the more drier conditions. This is what happens with, oh, I'm sorry. Yeah. This is what happens with the drought and the normal period. the process of flower initiation gets too much of scattered, which is also kind of confusing for the agents like co-evolutionary forces like pollinators. It's very, very confusing. You don't know when to go for the flowers or when you can. They think we can go on for the flowers every time. But this second peak in the normal years is quite normal for the anagysis because it has two bimodal kind of initiation of flowers. Basically, what happens with the drought is your normal life gets disrupted too much. And this is what happens with the fruits. Yeah. And then now if you look at the process, like how does this scatter? And also if you can now say whether this process have, in both during drought and the normal years, are they seasonal? In the sense, do they have some kind of a seasonality? Then you can use the circular statistics to measure that seasonality. And then you can see that in the abundant versus drought years. Okay, in all this, the seasonality of the process are significant in the sense they occur quite regularly or quite at the same time period of the year. Suppose now if you look at during abundant and normal years, the drought years, at the community level, the leafless stage is 36.0. That means most of the species remain leafless from the 36th day from January 1st. So that is the kind. But if you look at the normal years, okay, that becomes 39. That means there is a 3-day difference. The three days are advanced. Okay. And then most interestingly, if you look at the senescence also, it is 7.52. That is the 7th day from the January 1st. And it is like 10th day of the 10th day of the normal year, the January month, in the normal years or the abundant years. So there is a shift in the seasonality that we were talking about, what Dr. Singh has presented in the morning. And then if you now look at individual species, how does it matter? In the drought years, Anagysis latifolia has 127th day where most of the individuals of Anagysis is initiating leaves. That is 7 days after in normal years. During the normal years, it is 120th day that the Anagysis starts putting up the leaves. But in drought years, it is postponed by 7 days. We may think that, hardly it matters like 7 day, 3 day or anything. But for a tree at the community level, even a day shift is quite important. This is, there have been 100 years of monitoring in one of the wine yards in London or in England, so to say. And then they detected that the leafing process in that grapevine over the 100 years was pushed by 2 days. And then that had a significant impact on the. And then most interestingly, the Langstronia, which gets highly disrupted. During the drought years is the 152nd day, but in the normal years, it is 129th day. So there is a kind of real big shift among the Langstronia. This is what we may not be able to capture with the Phenocam. Because individual tree observations that we cannot capture, and then we cannot characterize that particular species. And that is why we need to have a ground data that validates our Phenocam data, and then we can draw inferences from that. And this is the, again, for at the community level, what happens during the drought years and during the normal years. It's all, I think, like push. Though in all these years, the seasonality is significant. That means during the drought year also, the flowers, fruits, and leaves happen at a very regular seasonal pattern. But only difference is that the days are shifted this way, that way, and that kind of shifts. If it happens quite frequently, then it will have a long-term impact on the tree's reproductive output or trees. What you say, growth of the tree, particular tree. This is what happens during drought and with the flower initiation. And this is the abundant at the fruiting at the community level. And this is what happens with the fruiting under different species. Between Anagasis, there isn't much difference. It just pushed by about 3 days. But you look at the Tectona, it was 272nd day, which is very early during the drought, but it has pushed almost to 318, which is almost close to the end of the year in the normal years. Similarly for Terminalia also. Yeah. There are two things that gets affected because of the drought. First is the seasonality, that is the shift in their days of fruiting and flowering. And the second one is the synchrony. The synchrony is the measure of like how do all individuals perform, say, the process at the same time, or they are kind of distributed. During, this is one of the important measures to look at how the impacts of a drought is on that. For example, now you look at 1 minus, I'm reporting the synchrony values, and 1 minus of that would be in asynchrony. So, for example, during drought years, Anagasis has 0.54. That means the level of synchrony is about 50%. And then during the normal years, it is 0.584. They're close to it. And most of them, for example, now you look at Tectona grandis is 528 and 579. The values are quite different. But the Terminalia is very, very significant. Like during drought years, not that, they are not very highly synchronous. There is quite wide variation among the individuals. But during the normal years, they quite become, all of them seem to be quite synchronously leafing out. This has a great significance because this also has an impact on its herbivory. If there is a large asynchrony present in the system, they are potentially, or they are they are exposed much more to herbivory damage rather than during the normal years, they aren't much, because they tend to, or they try to escape the herbivory. And then herbivory is a big phenomenon in which large part of the leaf lamina has been eaten, and that costs the productivity of the tree. So this is with the flower initiation, how this synchronous and non-synchronous is there, and this is with the fruits. Yeah. So what is the kind of take-home message from this drought and non-drought periods? First is the temporal scatter. That means the extension or presence of a particular phenophase at the temporal scale, the number of days, it's very high during the droughts compared to normal years. And then energy budget. I was talking about the energy budget. This is very, very important because if you are putting up your energy, or if you are spending your energy in putting up either leaves or flowers or anything, your production would get suffered. That in turns affects the carbon sequestration of the system. That happens during the drought. And seasonality is very strong in abundant years or the normal years compared to the drought years, and also synchrony. So all these things, seasonality, the synchrony, all these things in combination hits the plant on the energy budget, and it also reflects what was the productivity and what was the growth, and ultimately what is the kind of net carbon sequestration that is happening with that forest time. And we know that our systems, our dry forest systems, we are still kind of acting as carbon sequester. We are acting as a carbon sink rather than the source. So that would, if droughts happen, the kind of extreme events like droughts, if they have the frequency of its occurrence, if it goes on increasing, if the frequency increases, then the life pattern of these guys gets disrupted. And so ultimately our carbon sequestration potential comes down. That is the kind of impact on a longer or a larger scale the system or this one will have. Now, if you now want to see what would be the next, we can't just keep on just measuring or just noting down this is flowering, that is flowering, this is not flowering, that is not flowering. We need to go a step ahead and then see. In that sense, our direction of research or our direction of more understanding of these systems should go into the functional traits. We have to look into the differences between the canopy and the understory trees. In terms of its leaf, easy leaf area is much more in canopy species, or understories has more leaf area than the canopy. It is advantageous for the understory to have a larger leaf area compared to canopy. Is that present in the dry forest, or how is it affecting in the different forest types altogether? And then we also need to see how does this evergreen or the complete leaf forest is different from the deciduous. The deciduous trees have much more pronounced kind of different phenophases compared to the evergreens. Do the evergreens also has such kind of phenophases regularly happen? It does happen with my research in the upper mountain forests, and it is strongly affected. And then we also need to have some kind of a generalized phenological models, affecting, does moisture has a role in both evergreens and in the deciduous, because we are living in a seasonal country like us, like ours. We are not living in a seasonal country like the equatorial tropics. We are living in a seasonal tropics. So can we now say that in generalize the phenological impact, saying that if we have deficit or if we have some kind of imbalance in the moisture, would that affect the general phenology? And then that would result in the carbon sequestration pattern. So how much of the rainfall has, it's like understanding who is the immediate cause. For example, if you say that rainfall is an immediate cause, then we can also think of what is the force that driving the differences in the rainfall. Is the temperature is driving, or someone else is driving? Who is driving? Like that. And then we also need to have comparative phenological studies, in the sense, as much as possible, we need to look at the latitudinal differences. These kind of studies would definitely help us to sort of understand the general phenological patterns in India. So we are trying to move in that direction through our ICFR network, in which all the phenological monitoring is happening on uniform methods. So we also need to have satellite-based studies where we can generally look at indices like NDVI and EVI, understand the greening and the browning processes, and then we can corroborate these greening or the browning process with our ground observations. And then we also need to have Phenocams to understand the canopy phenology and the number of interannual variation in the number of leafy days in especially in the dry forest. And we account, India accounts about 40% of the Indian natural forests are deciduous. So deciduous forest has a very significant role in such understanding of our total carbon balance or total carbon budget. What I'm trying to say is that even though we have studies based on satellites, phenocams, we still need to have a strong ground-based study to calibrate and to understand the crucial interannual, interspecies, intertree differences. Then this network of phenocams is very, very essential. And what is the most important thing is we need to have many collaborative research on phenology, having a common protocol throughout India. That makes us a big, network like the Americans have in a network, and the Europeans also have a network. So that would build up the large, and we can answer several questions because of the climate impacts. And in this study, I would like to thank the following people and also agencies, Government of India, Ministry of Environment for their generous funding. And these people have helped me a lot. So thanks. And if any questions, I would like to answer.
I think the message is very clear.
Yeah.
It is really very important that the ground-based observations are primary. Everything else will build upon.
Exactly.
That message is very important. We have to get that message here. Also, sir, I wanted to check with you about your like one of the messages that you have given about who evergreen plots also have limitations. Because we are looking at, especially when we look at the Phenocam patterns, from evergreen plots, which is very difficult to classify the kind of S-shaped curve that Dr. Singh has shown. The start of season, end of season, middle of season, etc. In our place, we are able to get some of the signals because we have some deciduous species which are changing them. So I'm quite interested to understand what is that you are talking about. And you said that evergreen forests also show images. And is it possible to pinpoint that and try to identify that as a marker of
Beginning or end of the season?
yes?
Yes, very much possible. Even evergreen trees, even though they have leaves throughout their life or throughout the canopy or throughout the year, they still have their initiating phase, expansion phase, and then matured phase and senescent phase. They still have, and then each of these phase is significantly seasonal. That is where your ground observation comes. You can, our camera, our webcams or our satellite pictures may fail to capture that small variations, but our eyes, the ground, our eyes, we will not be deceived or we will not be deceived of taking that. So they do have, for example, if you take any of the Loracy members, the initiating leaves will be almost kind of reddish. And then as the leaves get expanding, as the leaves get elongated, so to say, then they start losing their pigments, anthocyanins, and they turn to be more and more greenish. Even in Oleandroyoga, where you will the dominant trees in Netravali, you can certainly make out the budding leaves And then you can certainly make out the expanding leaves or the elongating leaves sheerly by the difference in the greenness, color of the greenness. Like the expanding or the elongating leaves will have much lighter, like parrot green kind of a leaf compared to more mature, which is more darkish, the green side. So this kind of data is available, but I don't know, probably with the webcams, when we look at this different differences in the spectral signatures that each of these sending, then probably we may be able to pick up that. I'm not sure how best or how good that is possible.
Yes, that is actually point. Recently, in Odisha, where we have a Phenocam, we have captured this phenomenon. From satellite, we are not able to interpret that there is any distinct phenology cycle happening. But from the Phenocam, it has been captured because it is understory also is contaminating the signals on the satellite.
Yes.
And it is always showing as a reading. Where from the Phenocam, that even in the and we have found that this phenomenon.
Exactly.
It's a very incident observation that we have on the ground. And that's why not think is.
Sure.
This is one of the presentation that I but I have seen the forest papers and I have in the city. So I have seen that when the plot was established in Mumbai, there were 72 species in the entire, and probably there are 7,000 individuals that are placed on the probably.
26,000. 26,000.
I'm not accurate. But it has since then it has increased in terms of the species, it has increased 34 or so around 84. And the number of users have also increased to 25,000 or so. So does it mean that the climate change, the global change, whatever is happening is really like by the forest? Is the what I can see here, the species count is increasing in the given fixed area, and species individual members are increasing. So it is.
So
it looks like a very happy situation for the forest.
Correct.
So,
Correct. See, there are two. You can look at it in two ways. First one, the global impact or the impact of climate change on that forest. That is through the biomass. We have terrible disturbances like annual dry season fires, and then we have large mammals roaming around this forest. With all these things, we still have the biomass of the forest is going up. I think we started with 186 or 180 tons per hectare in 1988. Right now, I think we are somewhere around 224 tons a hectare. And that is largely contributed by growth of the hardwoods. So the question is, the biomass growth is moving from the softwooded trees to the hardwooded trees. We are losing the softwood trees. For example, one of the trees is Fibia. We have lost that. There is a terrible loss. Like we started with 5,000 individuals, and then at one point of time we came to something like 275 individuals. Now, because of the recruitment, we are still having something like 2,000 individuals.
What is the reason of loss?
The reason of loss is elephants. They browse on that, they kill on them. So the biomass, is shifting from the softwooded trees to the hardwooded trees, which is quite natural. In the sense, hardwood trees account for much more biomass, but the tilt in that, there is a shift in the balance, which may not be very good in terms of the general health of the ecosystem. That is one thing. But with the number of individuals going up, like we started with 26,000 individuals in 88. By 96, we came down to 15,000. From 96, we started some kind of going up, going down. From 2004 onwards, we started moving up. And that is largely because of the recruitment of an understory species called the Helictis. That's belonging to the Malvaceae.
But the species are also increased.
But those species numbers have increased and they are kind of coming, going. For example, Bruvia discon, which appeared in one census, it stayed for 2 censuses, it just vanished. That could be an accidental appearance and gone. But the more stabilized, more occurring species are about 70 or so. The remaining 80, we had about 84. I think our species standings is about 84 species, which came and which went, came and went, came and went. These kind of, they're not yet stabilized.
Can we say that some sort of carbon fertilization is helping the system, your ecosystem? Can we say? Because the system is very high.
See,
Carbon is not in species diversity.
Yeah.
Species individual count is increasing. What is what?
no, that is that is the. I'll tell you the other part of the story. For example, if you look at Anogenesis, which is one of the most dominant, one of the dominant canopy species, over the last 34 years, we just have had about 20 individuals which has recruited into 1 centimeter DBH class. And terminalia. Terminalia is even worse. We have just had about 10 individuals. But if you look at the initial size class distribution, when we established these plots, there was no individuals in the lower sizes. All of them are like greater than 5 centimeters DBH. That means at some point of time, these guys must have had recruitment, must have had 1 centimeter about. And then there is, now they have, I don't know what is the reason, they must have all the 1 centimeter fellows. Either they have grown up, there is no recruitment, something has happened. Thus, same thing, will it happen at some point of time for the forest to continue? That may not happen in my lifetime. It may not happen in your lifetime. Probably when our great-grandsons or great-grandchildren, when they come back to the forest and say, oh, this is where my great-grandfather had established a forest and he has observed this, probably then they might realize. This is the same thing that happened with the Helictis. No, we started with about 2,500 individuals in 1988. There was some kind of a decline. But from 2003, 2004 onwards, it started growing. By about 2008, it massively increased. And that is probably called the episodic recruitment, which is one of the adaptive features of the dry forest to maintain itself. That is like you wait for a favorable climate, favorable condition. Once you think that you have reached or you got that, you suddenly start expanding. The other way is like, do the species have some kind of a clock in them? And then say that, okay, boys, now we are in the stage of extinction. Now we should do, and we should put up all our offsprings. So let's put up all our offsprings and let them sprout and germinate. So even if 100 of them gets killed, there are still some 5 or 6 of them to survive in the forest. Is that happening? We'll have to see what is happening.
It's quite interesting.
Thanks. Yeah.
So about this recruitment, is it influenced by any other factors other than like ecology, like say some animals or anthropogenic activities or seismic rates are increasing there? Is there anything else?
See, recruitment of an individual is a very complex kind of a process. Now there are several ways it can be, it can happen. It can get hampered, so to say. Have you heard of the pre-zygotic, post-zygotic mortality? Okay. See, the recruitment, it can happen right at the tree level itself. In the sense, when the inflorescence is formed, And when there is mating happens and the seed is produced, seed may not develop at all. That is called the pre-zygotic mortality. And then even if it develops, it falls on the ground, it may not germinate because of several things. That is called the post-zygotic form of mortality. Even if you survive both pre-zygotic, post-zygotic mortality, there could be predators. They can come, they can chew up all seeds. And then it is not necessary that all predators, all dispersal agents are good enough. There could be some very beneficial dispersal agents. For example, you have heard of Amla, Philanthus. Philanthus has a dispersal agent called cheetal or the deer. So if you observe the Philanthus tree in the forest, whenever it is fruiting, you find many a times, not always, many a times, you find monkeys on the tree and then cheetal on the ground. There is a mutualistic interaction between cheetal and monkey, the langurs. So langurs drop the fruit, cheetals eat them little bit and then chew them off. So that helps the Philanthus seeds to break their seeds and they break the cover and then they grow or they germinate. That happens there. But there are like rodents, which generally feeds on everything and which clears up everything. So all these factors influence what the recruitment is. And then even if you cross all this and germinate, put up your own seeding, then what happens is there could be fire, which gets the top portion knocked off. And then in the dry forest, that is one of the important strategies to have a very good bottom, the ground or the root systems, very good root systems. So even though your top portion is gone, you are alive, and then you be present. So these are all some of the agents that hampers the recruitment process. With all these things, we calculate or we monitor the recruitment here for the individuals which come up to 1 centimeter DBH class, assuming that whenever an individual reaches 1 centimeter DBH class, it has a greater probability of survival. It survives. But even then, there could be a lot of mortality at the 1 centimeter DBH class. So it's not a very simple process that we assume or we think. It's really a complicated process.
So you also study these pre-zygotic and post-zygotic mortality and are like climatic factors strong in influencing mortality?
See, it may not be a very direct impact of climate on the pre-zygotic and post-zygotic. It could be a result of that. In the sense, I showed you, when the flowers initiation during extreme events such as droughts is kind of stretched. Okay, there is a large temporal variation, and then most of these species are bee pollinated. Bees have to go sit on the flowers, they pollinate. So if there is a climate change, and if there is very much, there is a large temporal scatter in that, in that particular phase, then bees might get confused. There will not be any big kind of, some bees may be theirs because it also has its own life cycle. Bees, everything will have its own life cycles. They may not be there, available for pollinating. So in that sense, even though you produce a lot of pollen, your pollen may go waste. You may not get pollinated. You may not get fertilized. So these are all some of the indirect effects on trees, and that would suffer and show up in the next progeny. That was what it happens. Yeah. Thanks a lot. And I'm here. If you have any doubts, we can always talk about it.
Thank you, Dr. Suresh, for sharing the observations that was made in Mudumalai permanent preservation plots. And also, we have a better understanding of various phenofaces and various forest types now, I'm sure. And this will help us a lot. And also elaborating more on how drought is actually impacting various phenofaces in our forests. And also one thing that I understood was also that field work is very important, because when you were mentioning about the loss in softwood and increase in the hardwood, we would relate it to climate change. Is it because of climate change? That's when Dr. Singh was also asking, why is it? So it confirms that a lot of field work is also equally important along with the technology that we are going to use. So we'll now break for lunch and we'll resume the session by 2:30. Yeah, thank you.